Welcome to this biochemistry lecture on amino acid biosynthesis. Today we're going to talk about how to manufacture, create de novo, basically, amino acids. In order to synthesize amino acids, we need a source of nitrogen, and in animals, glutamate and glutamine play that pivotal role. So the alpha amino group of most amino acids comes from a transamination reaction uh, where we transfer the amino group from glutamate to an alpha ketoacid receptor. In order to synthesize glutamate, though, that's synthesized from ammonia and alpha ketoglutarate by glutamate dehydrogenase. That's something you might recall from our earlier discussion of catabolism of amino acids. The reaction is on the top where you have ammonia plus alpha ketoglutarate plus NADH slash NADPH plus a proton, and that yields glutamate plus NAD or NADP plus and hydrogen. It's water, so it's a hydrolysis reaction. That's where nitrogen comes from. That's where the nitrogen comes from. The nitrogen comes from glutamate, and glutamate is made from ammonia and alpha ketoglutarate put together. That'll provide the backbone for all of amino acid generation. There's an interesting concept that's associated with amino acids in that we have essential and non-essential amino acids. Humans in particular, and this applies to most mammals, but we'll discuss it in terms of humans. Most humans cannot produce the essential amino acids, lysine, methionine, threonine, valine, leucine, isoleucine, phenylalanine, tryptophan, and histidine, and they can't produce them, so they need to consume them in their diets. And this shows, in addition, at certain times, things like asparagine, the body can't keep up with high demands, like, for instance, during adolescence, you have a hard time making enough asparagine to create all the necessary things. You need to supplement with your diet. And this chart shows all of the moles of ATP required to be produced in order to keep a cell functioning and healthy. So generally, we have essential and non-essential that are categorized, but you also have localized times when we have to consume more in our diet because we can't manufacture more. Again, so this kind of gives you an idea. Listing on the left is our essential and our non-essential amino acids. On the right, we have the pathway where we manufacture all these. And you can see from the table that this pathway is the amino acid biosynthesis pathways all center around glycolysis and into the citric acid cycle. So just to note, if we could take glucose 6-phosphate, turn it into ribose 5-phosphate in 5 steps, and then we can get from ribose 5-phosphate to histidine. If we're at the 3-phosphoglycerate step, we can manufacture serine, and from serine we can manufacture glycine and cysteine. At the 3-phosphoglycerate group, we can also create urethros 4-phosphate, and from there we can go to tryptophan, phenylalanine, and tyrosine. Or you can go to the next step in glycolysis, which is phosphoenol pyruvate, and we can go to those same targets. From pyruvate, we can go to alanine, valine, leucine, and isoleucine. When we get into the citric acid cycle, we can go from alpha ketoglutarate to glutamate, and from glutamate, we can go to glutamine, proline, and arginine. If we get all the way over to the other side of the cycle and we have oxaloacetate, we can generate aspartate, and with aspartate, we can generate asparagine, methionine, threonine, and lysine. So the general idea here is we can get to certain places as intermediates of cellular respiration cycles, which include the glycolysis pathway as well as TCA cycle. Again, this kind of gives you the common intermediates that we just talked about. Carbon skeletons of 20 amino acids are derived from just 7 metabolic intermediates, and those are found in 3 metabolic pathways. Again, the intermediates are 3-glycophosphorate, phosphoenol pyruvate, and pyruvate. We have 2 pentose pathway intermediates. That includes 5-phosphate and urethros 4-phosphate, 2 citric acid cycle intermediates, alpha ketoglutarate and oxaloacetate. And plants and bacteria are capable of synthesizing all 20 amino acid, but most animals can only do a restricted number, mainly the ones you see listed. That's an important consideration when we discuss about bioethicists or amino acid biosynthesis. So alanine and aspartate are essential, non-essential amino acids because we can make them from pyruvate and oxaloacetate respectively using a transamination. In contrast, you can see things like, and this is just a kind of a subtle but important point, things like methionine, histidine, and tryptophan, which are much more complex, we have a tough time making, although plants and animals make them, so we are fine. We generally lack the enzymes to manufacture them. In general, these structures that are essential are more complex than non-essential, and that's reflected in the number of enzymatic reactions required for synthesis. So the ones on the right that are essential are generally ones that we have to consume in our diet. They're also very difficult to synthesize, requiring multiple step enzymatic reactions. Again, the flip is also true to non-essential amino acids that you see on the left, alanine, aspartate, serine, where these things have simple enzymatic pathways, and they're relatively easy to manufacture. One of the other important concepts that we're going to discuss, that we need to discuss, is allosteric regulation of these enzymes. Amino acid groups have different chemical properties, and the necessary abundance of an amino acid is dependent upon the proteins that are being produced. So at certain times, certain proteins are being produced at larger numbers than other times, and because of that, it's not uniform. And because of that, we need to regulate it pretty tightly. So for example, lysine, alanine, valine, isoleucine, glycine, and glutamic acid are the most common amino acids, and therefore they need to be less regulated. I mean, we need upregulated more. They need to be activated, whereas cysteine, tyrosine, and histidine are relatively rare, and so we are going to need to take and regulate them when we go over abundance. And you can see in this chart things like if we're going to go from aspartate to aspartyl beta phosphate, there are multiple regulation points in there, including the A1 enzyme of this, lysine, will inhibit, as does isoleucine. Whereas A3, you can see that threonine regulates that. When we get from aspartate beta semialdehyde to a homoserine, all of the pathways are inhibited by lysine. Beta 1 is not inhibited, but beta 2 is inhibited by threonine and isoleucine, and so on and so forth. These are allosterically regulated enzymes, and it's very important to note that as we get in production, a high level of the molar concentration of these amino acids, you're going to see a more tightly regulated and inhibited biosynthetic pathway. Some of the other important concepts that we need to discuss. So bacteria and plants produce all 20 amino acids, whereas mammals and humans generally only produce 6. But there are intermediate pathways that and in bacteria there are intermediate pathways that interface between the two. So if we start with oxaloacetate and we do the aspartate aminotransferase gets oxaloacetate to aspartate. From aspartate, I can get to all of these different products. One of them having to be threonine. From threonine, we can go to threonine dehydratase that can make alpha ketobutyrate, and that pathway is tied in with pyruvate as it goes to isoleucine. So we have a tight intermingling of pathways that exist between all of these different amino acid enzymes and pathways. So the shikimate pathway is pretty important in plant amino acid biosynthesis. It provides a mechanism by which herbicides can be produced. So aromatic amino acids are synthesized in plants and bacteria by the shikimate pathway, and it requires the formation of a C10 compound called cortismate, and it's the precursor of all 3 aromatic amino acids, tryptophan, tyrosine, and phenylalanine. One of the most widely used herbicides is Roundup, and Roundup, also called glyphosate, which is the active ingredient, is a competitive inhibitor to the enzyme EPSP synthase, which converts the shikimate 3-phosphate to 5-enolpyruvyl shikimate 3-phosphate, and so it blocks the pathway. And since animals don't have this enzyme, it's generally regarded as safe herbicide in that pathway, and so it allows us to selectively apply. And obviously, if you can't produce nucleotides, you're not going to be around. Some of the inborn errors of metabolism are associated with amino acid biosynthesis. And the most common one, or probably the most prevalent one that exists, is PKU, phenylketonuria. Sorry, that one's just a tongue twister for me. I just call it PKU. So PKU is, there's an enzyme, phenylalanine hydroxylase, that takes phenylalanine and adds the hydroxyl group to it to make it tyrosine. And you can see that in this pathway. If you don't have that enzyme or don't have a functioning enzyme, you end up with PKU. Tyrosine is an important precursor to many of the neurotransmitters and other important hormones, including epinephrine, dopamine, and melanins. So that's really important as far as metabolism is concerned. And if you do not have PKU enzyme, then you end up with a disease state that's relative. I mean, people can survive with it. They just don't. They can't eat phenylalanine because they can't. One of the pathways and regulation mechanisms of phenylalanine is the production of tyrosine. And so, in PKU patients, you get too much phenylalanine and you get a toxic effect due to the overabundance of phenylalanine because you can't regulate it. And so, it's an important element in that disease state. This is a common disease state. There's several other ones we'll discuss more later. Appreciate you listening. I look forward to seeing you in the next lecture.